Well, dear students, today we will be discussing synthesis and catabolism of starch and sucrose. As you are well versed, during first unit, carbon dioxide assimilation, we have seen that during photosynthesis or during Kelvin cycle, both photochemical as well as the direct reactions, the ultimate product is the glucose. And it's this glucose which will be then in a stored form. That stored form is called as starch. So before we will understand the synthesis of starch, synthesis of sucrose, we would like to understand what is starch basically. Starch is a polymer of glucose which is linked together by alpha 4 glycosidic linkages. That means you have a glucose units, number of glucose units which are clubbed together by a 1,4 glycosidic linkage, which results in the form of a chain of glucose. That chain of glucose is called as the starch. And it is stored within the plastids or chloroplasts. Since it is a storage type of a polysaccharide, it is stored in the form of starch grains. And these starch grains during night in the leaves are acting as a source of sugars. If you will see, the starch is the most abundant carbohydrate, reserve food found in almost all plants. If you will take cereals, if you will take potato, you will take banana, you will have a product which is having these products have the abundant carbohydrate. Now, first of all, let's understand how these starch grains are basically looking. They're colorless, highly refractive in nature, having definite morphological features. They will have an hilum central point, which can be at the center or which can be away from the center, and they have certain striations. When we see morphologically, they have a shape, size, which is always unique to a particular plant species, and it's also used as a morphological identification marker. Now, having said that starch is a stored form of a polysaccharide and is stored in the form of starch grains, you will see these starch grains are basically having definite shapes. If we will consider the starch grains of cereals, which includes the in cereals as well as in potato, you will see potato has a starch grain of this shape, which is having an eccentric hilum and is having in concentric striations. Likewise, wheat starch grain appears like this. It will have an hilum like this, which can be at central. Or in case of maize starch grain, you will have a cleft form of a triradiate ridge hilum. But in case of cereals, in oat, in rice, as we have seen in case of the rice, you will see there are the compound grains or there can be simple grains. So that means it is a form of a stored polysaccharide. When we discuss the chemical nature of these starch grains, you'll see starch is composed of two polysaccharides, namely amylose and amylopectin. What is the difference between the two? Amylose is an unbranched helical molecule composed of about 200 to 2000 alpha D glucose units joined together by an alpha 1,4 linkage. That means amylose has a number of D glucose units, which can be from 200 to 2000, but they are linked by means of 1,4 linkages, and it is unbranched. And you will see because of the 1,4 linkages there, it will be in the form of an helix because it assumes a bend due to 1,4 bonds, which results in the form of a hollow helix. That's why this hollow helix gives it starch a less fibrous quality and more granule-like shape, best suited for storage. So it is their chemical nature which is making it a less fibrous material and suited for storage. And this amylose, when you see, due to simple structure, is more soluble in water. It turns blue black in color after treatment with iodine solution. If you have to identify whether a material contains a starch, you have to add an iodine, and because of its chemical nature, it turns blue black on addition of the dilute iodine. Having said, what is the difference between the amylopectin? Well, at the same time, amylopectin is much branched, with main chain composed of alpha D glucose units, same as amylose, but they have the side chains after every 30 glucose units with alpha 1 6 linkages. That means the difference between the two lies here. That's a straight chain. It has a side chains. The side chains are at 1 6 linkages, alpha 1 6 linkages, which can be after 30 glucose units. So it is complex structure. Amylopectin is a complex structure. Thus, it is less soluble in water. And its coloration on iodine, it turns red to purple color. Now we'll see them chemically, how they are looking. Amylose, as I said, has a chain of D-glucose units which are joined by means of 1, 4 alpha linkages. One of this and the 4 of this forms alpha linkage. So there are a number of these units. And since there is a bend here, it assumes the shape of a helical form, and so is a hollow helix. That's why it is in the form of starch grains, which is a storage product. In case of amylopectin, you have a main chain that of course looks like the amylose, having the same 1,4 alpha linkage, but there are side chains which will be branch chains. Side branches will be 1, 6. So this is a structure of an amylopectin and amylose. Coming to the starch synthesis, let's understand where it is being synthesized. Since we know it is a photosynthetic product, it has been produced by means of the photosynthesis in the leaves. So starch is synthesized within the chloroplast, no doubt about that. So we have seen in case of carbon dioxide assimilation that starch synthesis or glucose synthesis takes place within the chloroplast, and that too the stroma of the chloroplast, which is a dark reaction. So starch is synthesized within the chloroplast, and starch is temporarily stored inside the chloroplast. And let us understand, before we will go the regulation of the starch synthesis, I'll just to give an idea that it is basically when in case of Calvin cycle, during dark phase, which has several three phases. One is the carbon dioxide fixation, and then reduction and the regeneration. So carboxylation phase, and then reduction phase, and then there is a regeneration phase. But out of this Calvin cycle, what you get? You get a glucose. That glucose is then further processed for the starch formation, where there is a enzymes which are responsible for it, where more energy will be used. We have a glucose 1,6-phosphate, which is the outcome of the Calvin cycle, that in presence of ATP and in presence of a key enzyme, ADP glucose pyrophosphorylase, it results in the formation of ADP glucose units, and there is also the formation of the pyrophosphate. And what will happen to the pyrophosphate? That's a separate story. A pyrophosphate in presence of pyrophosphatase and water, it will be changed back to the 2 orthophosphates and 2 protons. But what will happen to this AD glucose? It is this AD glucose molecule which in presence of a starch synthase will form the ADP will be out, and there will be the formation of a starch. Is a precise that how starch is synthesized. That means the basic units for the starch is our glucose units, and which is with the help of this key enzyme, it results in the formation of ADP glucose, and then there is a formation of a starch. Before we have seen the structure of the starch, now we will understand the synthesis of the sucrose. As you know, sucrose is also a photosynthetic product. It is a transportable polysaccharide which is synthesized in the cytosol of the mesophyll cells. Starch is synthesized within the stroma of the chloroplast, and sucrose is synthesized within the cytosol of the mesophyll cells. What is basically a sucrose first? It is a heterodisaccharide. It's made up of two types of the monosaccharide units, D-glucose and D-fructose, which are joined by means of 1,2 glycosidic linkages. So there are two types of, one is a type of a pyranose type of a ring, another is a fructofuranose type of a ring. So we have sucrose now. In case of plants, it's considered as a major carbohydrate produced by plants and is transported to other parts of the plant to storage organs through phloem. Sucrose, in presence of enzyme sucrase, is hydrolyzed to glucose and fructose residues. Now let us understand what is the chemical structure of the sucrose. Chemical structure of sucrose is it has two units linked by means of 1,2 linkages. One is D-glucose, and one is fructose. So let us understand in brief how this sucrose is synthesized within the cytosol of the mesophyll cells. Then we will discuss its regulation also. As you see, it is produced by condensation of UDP glucose and D-fructose, which is regulated by phosphorylation of sucrose phosphate phosphatase, which is an enzyme in the mesophyll cells. It is synthesized within the cytosol of the mesophyll cells. This is a summary, and we will be discussing it in the detail. You see, we have, in case of the mesophyll cells, through one of the pathways, you are getting the glucose 1-phosphate, and then in presence of UTP, uridine triphosphate, in presence of this enzyme, which is UDP glucose pyrophosphorylase, there's a formation of UDP glucose, and there is also pyrophosphate. Then what will happen to the pyrophosphate, which is taken place in the later on? Whereas UDP glucose will be combining with the fructose 6-phosphate. As a result, there will be two units of it in presence of an enzyme, sucrose phosphate synthase, and it results in the formation of sucrose 6-phosphate, releasing UDP. And this sucrose 6-phosphate, in presence of water and in presence of a sucrose phosphate phosphatase, it results in the formation of a sucrose and releasing orthophosphate or inorganic phosphate. In a nutshell, we are discussing the synthesis of the sucrose, and mind you, it's taking place within the mesophyll cells. That means we have two processes. One is the starch synthesis, another is the sucrose synthesis, and both are taking place in the two compartments of the cell. One is in the chloroplast stroma, another is in the cytosol of the mesophyll cells, and both are competing processes, both are competing reactions. They are competing each other. We'll see through this diagram how it's being synthesized. See, this is the chloroplast. This is the cytosol of the mesophyll cell. We have the center of it, the Calvin cycle. During daytime, Calvin cycle, you have understood, there is carboxylation, reduction, and formation of hexose and regeneration of ORABP, but there are formation of triose phosphates. Multiple triose phosphates are produced there. They will undergo a set of reactions. Through these reactions, the ultimate product, which will be the production of the hexose, that is glucose 1-phosphate. This glucose 1-phosphate, in presence of an enzyme, ADP glucose pyrophosphorylase, it requires an ATP and releases pyrophosphate and forming an ADP glucose, which will later on form the, in presence of this enzyme, Starch Synthesis. As I have given the summary in the earlier slide, you will see the synthesis of the starch. So mostly we will see the starch will be produced during the daytime, because during daytime there is tremendous production of triose phosphates, which of course will be reduced, of course will be resulting in the formation of glucose, and the glucose will be converted into starch. But there is a communication between the two, that is this chloroplast and the cytosol. Here is another reaction which is taking place for the synthesis of the sucrose. So we have two compartments of the cell, chloroplast and we have the cytosol, where between the two, in the membranes of the chloroplast, there are present certain translocators. They are called as orthophosphate, triphosphate translocators. They are basically antiporters. That is, they will strictly pass on from one side phosphate, and from the other side they will be transporting the triose phosphate. Let us understand in detail what is happening. We'll come to this slide again. We have a regulation of starch and sucrose synthesis. Synthesis of starch and sucrose are two competing reactions, as I have said. That is, these reactions are competing each other, and the two compartments of cell communicate with one another through orthophosphate, triophosphate, triphosphate translocators, or which is an antiporter. This is the chloroplast membrane. In between them is present this antiporter, which is the cytosol side. This is the stroma side. The inorganic phosphates will be taken from cytosol to the stroma of the chloroplast, while as the triose phosphates will come out in exchange through these into the cytosol. So that means there is a communication going on between the two. Now, how it is being regulated? Now, we have understood that phosphate translocator, which is located in the membrane of the chloroplast, catalyzes the movement of orthophosphate and triose phosphate in opposite directions between chloroplast and cytosol. That means from cytosol it will take the phosphate inside, and from the stroma or from the chloroplast, the triose phosphates will come out of it. Relative concentration of, now what is the triggering point? What will be the factor responsible for the movement of this? That is the relative concentration of orthophosphate and triose phosphate. That will determine the synthesis of starch or sucrose. It will determine the synthesis of starch or sucrose. If we have a low concentration of orthophosphate in cytosol, it will limit the export of triose phosphate. That means if there are less phosphates, orthophosphates in the cytosol, they will not enter, there will not be the exodus of triose phosphate. Thus, it will promote synthesis of starch inside the chloroplast, because if triose phosphate is not coming into the cytosol, it will be used for the formation of the starch in the chloroplast. If we have an abundance of orthophosphate in cytosol, what will happen? It will have an opposite effect. There will be now synthesis of sucrose. It can be cleared by this slide. We have the two situations here. In one situation, we have a low level of orthophosphate in cytosol, where in other situation we have an abundant orthophosphate in the cytosol. What will happen when there is low? There will be no exchange taking place. No exchange. That means in chloroplast, the Calvin cycle trioses through a process of reactions, trioses, there will be the synthesis of starch. So low level of orthophosphate will result in the synthesis of starch synthesis of the chloroplast. Now, in case if there is an abundant inorganic phosphate present or orthophosphate present in the cytosol, what will happen? That will go through the antiporter inside and triose phosphates will come out. And those triose phosphates will be utilized for the synthesis of sucrose. That means there will be the synthesis of sucrose which can be taking place through these set of reactions now. That is triose phosphate. It will be taken inside from chloroplast to the cytosol, and here through these reactions there will be formation of fructose 6-phosphate that will form the glucose 6-phosphate, and that through these reactions and with the UTP and this fructose, there will be combination of the two that will form the sucrose phosphate, and the phosphate will be released and there is a synthesis of sucrose. That means if there is triose phosphate available, that will be utilized for the formation of this sucrose. Now, let us understand this orthophosphate and triose phosphate. Now, it seems that it controls the activity of several regulatory enzymes in the sucrose as well as the starch biosynthetic pathways. And you will see a situation during light and during dark. What will happen? We have a key regulatory enzyme. That key regulatory enzyme in starch synthesis is ADP glucose phosphorylase, which is a key regulatory enzyme, which is responsible for glucose 1-phosphate with ATP forms ADP glucose, which are the basic residues of the starch. What will happen during the night, during light, and what will happen during the dark? Let's understand first what will happen during the light. During light, active photosynthesis is taking place, and carbon dioxide assimilation is also taking place there. There is more phosphoglycerate formation, that is trioses are produced there. And these trioses will stimulate this ADP glucose phosphorylase. As a result, there will be the formation of ADP glucose. That is during light. During dark, what will happen? There will be no photosynthesis taking place. There will be more availability of pyrophosphates, these inorganic phosphates or orthophosphates during night. And they have an inhibition. They will cause an inhibition on this enzyme. So it will be inhibited during night. During dark, there will be no ADP glucose and there will be no starch synthesis. That means during light and during dark, it is a key enzyme which is being regulated. That is called as a ADP glucose phosphorylase. Now, let us understand, we have seen how starch is being regulated. So in case of starch, you have seen that the key enzyme for its regulation is that is this enzyme, ADP glucose phosphorylase. Now, during day, during night, how it's being regulated. Now let's understand how there is a regulation of regulation of this sucrose synthesis. The structure of the sucrose. let us understand and the brief brief synthesis of the sucrose as it is taking place inside the mesophyll cell cytosol. How it is regulated? Synthesis of sucrose is regulated by phosphorylation of sucrose phosphate synthase. And there is another molecule which is called as fructose 2,6-bisphosphate, which acts as a key control molecule that allows increased synthesis of sucrose in the light and decreases synthesis in the dark. Fructose 2,6-phosphate is present in the cytosol. You should not confuse fructose 2,6-bisphosphate with fructose 1,6-bisphosphate. These are two different molecules. So they are in minute concentrations and they exert a regulatory effect on the cytosolic interconversion of fructose 1,6-bisphosphate and fructose 6-phosphate, because these two things are very important. Fructose 1,6-phosphate will be converted into fructose 6-phosphate, and fructose 6-phosphate is a basic ingredient for the synthesis of sucrose in mesophyll cells. So what is happening basically here, you'll understand that a fructose 2,6-bisphosphate, when it is present, it exerts a regulatory control. One way, see, triose phosphate will be converted into fructose 1,6-bisphosphate, 1,6-bisphosphate to fructose 6-phosphate, and that is the basic thing for the sucrose. Or through another enzyme, it will be converted into fructose 2,6-bisphosphate. Now what happens here? Regulation is this way. This fructose 2,6-bisphosphate exerts an inhibitory control on fructose 1,6-bisphosphatase. And that means when this fructose 1,6-bisphosphatase is inhibited by this, there will be no conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. That means there will be less availability of fructose 6-phosphate and there will be less sucrose synthesis. At the same time, it has a stimulatory role on this phosphofructokinase 1. Having a stimulatory control on it, it will allow the fructose 6-phosphate to be changed to fructose 1,6-bisphosphate. Again, there will be less availability of fructose 6-phosphate and less synthesis of the sucrose. And that means we have seen that increased cystosolic fructose 2,6-bisphosphate is associated with decreased rates of photosynthesis. And when fructose 2-bisphosphate is present, it acts as a powerful inhibitor of cystosolic fructose 1,6-bisphosphatase and an activator of pyrophosphate dependent linked phosphofructokinase 1. In both the cases, as I have shown here, in both the cases, it will result in the less synthesis of fructose, less availability of the fructose. So when there is less availability of fructose, there will be less synthesis of the sucrose. Likewise, low systolic ratio of triose phosphate to orthophosphate promotes formation of fructose 2,6-bisphosphate. And this formation, fructose 2,6-bisphosphate, is regulated, its concentration is regulated in light. And it controls the concentration of fructose 2,6-bisphosphate. Light controls the concentration of 2,6-bisphosphate, which has a regulatory role on the synthesis of sucrose. Well, my dear students, through this video, we have seen that what is basically starch, where it's produced, and how it is a storage product. And then we have seen what is the possible synthetic pathway of this starch. And we have seen that the starch is synthesized within the stroma of the chloroplast. And then we have seen that Its structure is such that it acts as a storage polysaccharide. And likewise, we have seen the sucrose, which will be produced within the mesophyll cells, and which of course is also regulated by key regulatory enzymes, and certain molecules act as a powerful regulators for it. As a result, in the two compartments of the cell, an exchange is taking place. That is, there is an exchange of pyrophosphate and triose As a result, sucrose will be synthesized. So that means we have seen synthesis of both starch and synthesis of the sucrose. Thank you very much.